Hello, my name is Sarah King, and I'm a PhD student studying geology at UC Davis. And I'm currently working with Dr. Don Sumner and Dr. Xiaoli Dong on understanding self-organized microbial patterns in Antarctic lakes. So in particular, I'm interested in understanding how climate change is impacting these kind of sensitive Antarctic lake ecosystems. And I am looking at Lake Vanda, which is a lake in the McMurdo Dry Valleys of Antarctica, which has experienced over 10 meters of lake level rise from the 1980s to 2016. And Lake Vanda has benthic microbial mats largely comprised of cyanobacteria spanning across the majority of the explored lake floor. And since these microbial mats are the dominant life form in this kind of extreme cold ecosystem, this raises the question, can we use these microbial mats to understand climate change? So in order to answer some of these questions, we send the cold-tolerant members of our lab down to Antarctica to collect image data of the microbial mats, which can then be converted into 3D models using a process known as photogrammetry. Once we have these models, we can perform spatial statistical analyses to understand how the pinnacles you can see present in the models are shaped and how they are spaced relative to each other. So one such metric we can use to quantify the spatial patterning of these pinnacles relative to each other is called the Ripley's L function. So you can see in these graphs a variety of gray envelopes, and these gray envelopes represent complete spatial randomness. And if the measured values taken from the microbial mats are above the envelope, then the pinnacles are clustered. If they're within the envelope, the pinnacles are randomly spaced. And if they are below it, then they are regularly spaced relative to each other. So using this metric, we can essentially see that there is a wide variety of spatial patterning present in the lake. So our next steps are to understand why there is a wide variety of spatial patterning in the lake. And we'll be using Bayesian causal modeling to quantify the degree to which each environmental variable influences pinnacle morphology. Since these environmental variables are all in turn impacted by climate change, this could ultimately give us the ability to predict how climate change will impact these microbial mats in the future. Thank you so much to the ARCS Foundation for supporting my work. Thank you for listening to my talk. And I hope to see you at my poster in the future so we can chat a little more about my research.